Hello everyone, now let us discuss about Good Manufacturing Practice Guide for Active Pharmaceutical Ingredients, Part 13. This comes under Q7 Quality Guideline of ICH. First, we'll be discussing about the APIs for the use in clinical trials. Coming to the general guidelines, not all the controls in the previous sessions of this guide are appropriate for the manufacture of a new API for investigational use during its development. That is, APIs for clinical trials. The controls used in the manufacture of APIs for the use in clinical trials should be consistent with the stage of development of the drug product incorporating the API. And the process and the test procedure should be flexible to provide scope for the changes as knowledge of the process increases and clinical testing of the drug product progresses from preclinical stages through clinical stages. Once the drug development reaches the stage where API is produced for use in drug products intended for clinical trials, manufacturers should ensure that APIs are manufactured in suitable facilities using appropriate production and control procedures to ensure the quality of the API. Coming to quality, appropriate GMP concept should be applied in the production of APIs for the use in clinical trials with the suitable mechanism of approval of each batch. A quality unit independent from production should be established for the approval or rejection of each batch of API for the use in clinical trials. Some of the testing functions commonly performed by the quality units can be performed within other organizational units also. The quality measures should include a system for testing of raw materials, packing materials, intermediates and APIs. Process and quality problems should be evaluated. Labeling for APIs intended for use in clinical trials should be appropriately controlled and should identify the material as being for investigational use. Coming to equipments and facilities. During all phases of clinical development, including the use of small-scale facilities or laboratories, to the manufacture of batches of APIs to use in clinical trials, procedures should be in place to ensure that equipment is calibrated, clean and suitable for its intended use. Procedures for the use of facilities should ensure that materials are handled in a manner that minimizes the risk of contamination and cross-contamination. Materials should be handled such that they must minimize the risk of contamination and cross-contamination. Coming to the control of raw materials. Raw materials used in production of APIs for the use in clinical trials should be evaluated by testing or received with the supplier's analysis and subjected to identity testing. When a material is considered hazardous, a supplier's analysis should suffice. In some instances, the suitability of raw material can be determined before use based on acceptability in small-scale reactions rather than on analytical testing alone. Coming to production, the production of APIs for use in clinical trials should be documented in laboratory notebooks, batch records, or by other appropriate means. And these documents should include information on the use of production materials, equipment, processing, and scientific observations. Expected yields can be more variable and less defined than the expected yields used in commercial process. Investigation into yield variations are not expected. Coming to validation. Process validation for the production of APIs for the use in clinical trials is normally inappropriate, where a single API batch is produced or where process changes during API development make batch replication difficult or inexact. The combination of controls, calibration, and where appropriate, equipment qualification assures API quality during its developmental stage or phase. The process validation should be conducted when batches are produced for commercial use. Even such batches are produced on pilot or small scale. Even if such batches are produced on pilot or small scale, the process validation should be conducted when the batches are produced for commercial use. Coming to changes. Changes are expected during development as knowledge is gained and production is scaled up. Every change in the production, specifications or test procedures should be adequately recorded. Coming to laboratory controls, While analytical methods performed to evaluate a batch of API for clinical trials may not yet be validated, they should be scientifically sound. A system for retaining reserved samples of all batches should be in place, and this system should ensure that a sufficient quantity of each reserved sample is retained for an appropriate length of time after approval, termination, or discontinuation of an application. Coming to documentation, a system should be in place to ensure that information gained during the development and the manufacture of APIs for use in the clinical trials is documented and available. A system should be in place so that all the development and manufacture of API used in the clinical trials is documented and available. The development and implementation of the analytical methods used to support the release of a batch of API for use in clinical trials should be appropriately documented. A system for retaining production and control records and documents should be used, and this system should ensure that records and documents are retained for an appropriate length of time after the approval, termination, or discontinuation of an application. Thank you for watching. Please subscribe for further videos on pharmaceutical sciences and other related disciplines.